Meet the Scientist webinar series. I'm Dr. Jeff Borenstein, President and CEO of the Foundation, and your host and moderator for today's webinar. Today, Dr. John Rubinstein will present a webinar entitled Understanding Brain Development as a Foundation for Understanding Brain Diseases. The Brain and Behavior Research Foundation is dedicated to funding research around the world to identify the causes, improve treatments, and ultimately develop cures and preventative techniques for mental illness. The Foundation has awarded over $300 million in research grants in a little over the past 25 years. I'm delighted to introduce Dr. John Rubinstein. Dr. Rubinstein is the Nina Ireland Distinguished Professor in Child Psychiatry at the Nina Ireland Laboratory of Developmental Neurobiology in the Department of Psychiatry at the University of California in San Francisco. Dr. Rubinstein's basic research interest is in developmental neurobiology, particularly the development of the forebrain, the area of the brain that is required for the highest intellectual functions. His clinical interest is in neurodevelopmental disorders. Dr. Rubinstein trained at Stanford University and the Pasteur Institute in Paris prior to joining UCSF. While serving as a resident physician at Stanford, he began experiments on the genetic control of forebrain development that remain a focus of his work at UCSF. Dr. Rubinstein has been a member of the Brain and Behavior Research Foundation Scientific Council since 1998 and is a 3-time NARSAD grantee. This will be an interactive event. We'll start with Dr. Rubinstein's presentation, followed by a question and answer period. To submit your questions, please use the Questions tab of the webinar control panel. You can submit your questions throughout the presentation. As your moderator, I will present your questions to Dr. Rubinstein, and we will address as many of them as possible. And now, I'm pleased to present Dr. John Rubinstein. John, the floor is yours.
Thank you very much, Dr. Borenstein. Can everyone see my presentation now?
Yes.
Okay.
Yes.
All right. I'm a developmental biologist who focuses on understanding how disruption of brain development can lead to mental disorders. What I'm going to do today is give a general introduction to the multiple steps that are required to form a healthy brain. Many of these, or most of these, take place while the baby is still developing in utero, and some of them take place postnatally through early childhood and into adolescence. I think, and I believe many scientists believe, that genetic and non-genetic disruptions of several of these steps may be the etiologies for mental disorders. Therefore, to try to understand how this happens, one needs to have a good foundation in the fundamentals of how the brain develops. So that's what we're going to do in this talk. The overarching hypothesis that my laboratory tests is that many psychiatric disorders are caused by dysfunction of brain circuits due to abnormal development and or function of specific circuits. So in thinking about brain development, we need to think about the components that generate circuits, and then later the molecular mechanisms that regulate the activities of these circuits. In the brain, you can think of two simple but general types of circuits. One would be a long-distance circuit, as shown in this picture. In this example, neurons in the cerebral cortex send their axons, which are colored in green in this picture, to neurons that are housed in a nucleus called the striatum. Then in the striatum there are two major kinds of neurons, ones which are called direct neurons and the other ones that are indirect, shown in red and blue respectively, that then complex connections into deeper parts of the basal ganglia, eventually going to the midbrain. I'm not showing all of this circuit. It gets more complicated as one looks at it in more detail, but this is an example of a long-distance circuit whose function is absolutely essential for our mental functions. On the other hand, there are circuits called local circuits, and here we're looking at a very schematized view of a local circuit that's present in the cerebral cortex. So in this case, it's important to understand that the cerebral cortex has two kinds of neurons, ones that are excitatory and others that are inhibitory. An excitatory neuron will excite other neurons, whereas an inhibitory neuron will inhibit the activity of other neurons. In this picture, the excitatory neuron is orangish red. There's only one excitatory neuron in this picture. And all the other neurons in this picture are inhibitory, and they are synapsing on to the excitatory neuron in different places. So for instance, this blue basket cell will synapse onto the cell body of the excitatory cell. Notice that the excitatory cell shape is triangular, and for that reason, excitatory cells in the cortex are often called pyramidal cells. So this basket cell synapses on the cell body where it inhibits the activity of the pyramidal cell. Another kind of inhibitory cell, like this green one, will synapse on the dendrite of the excitatory cell and inhibit the dendrite. The dendrites you can think of as being the antennas for the excitatory cells in which they receive information, whereas the axon, which is coming out at the bottom side of this excitatory cell, is the output for the information that the excitatory cell sends to other neurons. So this is an example of a local circuit that takes place in the cerebral cortex, and it controls the balance of inhibition and excitation within the cortex, a balance whose function has to be tightly tuned in order to correctly control neuronal activity in the cortex. So now I'm going to review major steps in brain development that are required to make these local circuits and long-distance circuits. So here are a number of steps that take place sequentially during development of the brain. I'm going to go through very briefly each of these steps. I'll name them now. First one is neural induction. It takes place just a few weeks after fertilization of the embryo, and it's a process in which stem cells of the embryo are converted into the neurons of the embryo. The next step is called neurulation, and that's where the primitive nervous system forms a tube called the neural tube. Then a process called regional patterning takes place, in which the embryonic brain is subdivided into its constitutive parts. And proliferation is a process that creates enough of the cells that are required to generate all of the neurons and glia of the brain. Migration is a very important process in which cells that are produced in one place migrate to a destination which is often quite far from the source of their production. Then, as I mentioned, there are axons which come out of the neurons, and they have to grow to the right place where they synapse on the appropriate cells. And then the glia, including the myelinating glia, will form insulations around the axons that increase the rate of their signal transduction. So I'll start by telling you a little bit about neural induction and neurulation. So in this picture, we see three schemas. On the upper left is the neural plate, which is the primitive neural ectoderm, which is formed during the process of neural induction. And in the primitive neural plate, there are already different subdivisions. The red subdivision in the middle is called the floor plate. That will eventually become the ventral midline of the neural tube. And at the lateral edges of the neural plate in lavender is the onlage of the roof plate, which will become the dorsal midline of the neural tube. Morphogenesis of the neural plate into the neural tube is shown in two steps to the right. The first one, there's a folding of the lateral edges of the neural plate towards the dorsal midline. They have not yet fused. And then in the bottom right, you have a fusion of the lateral edges. Those are the lavender color to form the roof plate. Disruption of this process, as probably many of you know, leads to holes or gaps in the dorsal part of the neural tube. And unfortunate patients who have this problem will have, if it happens in the spinal cord, something called spina bifida. If it happens in the brain, this process, this state, however, does not allow the baby to live, and they'll have a severe neural defect called anencephaly. Once neurulation forms the neural tube, the different subdivisions of the neural tube have to be established. And part of this takes place through structures called patterning centers that are present in the dorsal midline, in the roof plate, at the front of the brain, and also in ventral regions. Patterning centers at the dorsal part are shown here in green, and they produce molecules such as BMPs and WNTs, which are molecules that promote dorsal identity. At the front of the brain is a rostral patterning center, here labeled in blue by the letters CP for commissural plate. And this rostral patterning center is essential for creating the identity of the most anterior structures of the brain, including the prefrontal cortex, which is, of course, extremely important in higher level thinking. And then the ventral regions are patterned by molecules such as sonic hedgehog or SHH. These red regions that I'm pointing to are the ventral regions where sonic hedgehog promotes the identity of the hypothalamus and the basal ganglia. Mutations in genes such as sonic hedgehog or FGF8 cause severe problems in the development of the brain. For instance, sonic hedgehog mutations in humans leads to a very sad disorder called holoprosencephaly, where there's a very severe dysmorphogenesis of not only the brain but also the face. And the next step we'll talk about is how regional patterning is established along both the front to back, which is the anterior-posterior axis, and the top to bottom, which is the dorsal-ventral axis. What we're looking at here is not a map of Manhattan, but a map of the embryonic brain. Just like a street plan in a city like New York, there is a very specific plan that the development of the brain establishes to form specific structures at specific locations. I know this is much more complicated than you all want to listen to. I just want to point out some features of this. Where I'm pointing to with the arrow currently is the rostral or anterior end of the brain, and this is where your forebrain will be established. And the cerebral cortex, which I've been talking to, is just this little purple area that I'm circling currently. In the pre-plan of the brain, the eye, for instance, has a very large kind of surface area, whereas our cerebral cortex, which is much, much bigger than the eye later on, is relatively small. The main point to understand here is that there are molecular mechanisms that establish this exquisitely specific control of regional identity in our brain at very early steps. And mutations and other variables that alter this can change in major ways the nature of our brain subdivisions and their connections. When this neural plate stage fate map folds into a neural tube shown here, we can see the forebrain, which consists of the telencephalon, hypothalamus, and diencephalon formed. We'll be talking much more about the telencephalon, which consists of the cerebral cortex and the basal ganglia. But of course, all these other regions are extremely important in understanding the neural circuits that control how our brain functions. Many of you probably know that psychiatric medicines that are prescribed often affect the functions of neurons that use monoamine neurotransmitters, such as serotonin, which is probably the most famous. It's interesting to note that the positions in this embryonic brain that produce neurons that are capable of making these monoamines is highly restricted. So for instance, serotonin neurons, which are red in this case, the ones that are most important for regulating the activity of our telencephalon, these neurons are located just here in the anterior pons, in the median and dorsal raphe nuclei. Other neurons that are very important in our brain function are the dopamine neurons, which are shown here in yellow. And they are only located in the ventral parts of the midbrain, the substantia nigra and ventral tegmental areas. It's their axons from these serotonin and dopamine neurons that then grow long distances and then innervate the neurons here in the telencephalon in our forebrain that are most relevant for understanding how these neurons regulate our behavior and how medications that we use regulate the activity of signaling from the dopamine neurons and the serotonin neurons. Okay, let me segue now to another step in neural development, which is proliferation of cells. In this case, we call them the stem cells and the progenitor cells that are required to make all of our neurons and all of our glia. This is a frontal section through the embryonic telencephalon. We have the cerebral cortex, where my arrow is cursing now, and then the basal ganglia. In the center, this white area is the ventricle of our brain, and that's filled with a special fluid called the cerebral spinal fluid, which is, you can think of it as the nutrient, carrying the nutrients for our developing and our mature brain. Adjacent to the ventricle, where this rectangle is drawn, are where the stem cells are and the progenitor cells. So if we go from panel A to panel B and look schematically at the dividing cells. The stem cell zone, which is called the ventricular zone because it's next to the ventricle, contains a specialized kind of neuron called a neuroepithelial cell, also sometimes called a radial glial cell, and it's shown here at different stages of cell division. The M is the mitosis phase of cell division, and then as the cell leaves M phase, it goes through G1 phase, and then S phase. S phase is where the DNA replication takes place. And then the cell cycles back to a G2 phase, and then back to M phase where cell division takes place to produce two neurons. And this is the characteristic mechanism of cell division of these stem cells in the ventricular zone. Once cell division happens, these cells can either migrate into the subventricular zone, which is a secondary progenitor zone, where proliferation continues. That's shown by arrow 2, or they can migrate, as shown in arrow 3, into the mantle zone, which is where the neurons migrate after they have finished dividing. Let me just point out that in the subventricular zone, the secondary progenitor zone, the cell division gymnastics of these cells is very different than in the stem cell zone, as shown by the cartoon, that the shapes of the cells are very different at different, if you compare the ventricular zone and subventricular zone. In the human brain, the subventricular zone becomes extremely large. I'm now going to return to patterning of the brain and discuss how transcription factors control the identity of the stem cells that we were just talking about. So in panel A, we see this frontal view of the telencephalon again. The cortex is where my arrow is, and it's in purple and blue, and orange and red. And the basal ganglia is in green, yellow, and light blue. If we take these stem cells that are shown here in panel A and stretch them out in a line in panel B, we can see the stem cells in the ventricular zone and then the secondary progenitor zone or subventricular zone, shown as these two elongated rectangles. The organization of these progenitor zones is schematically described here. The purple part is the most dorsal part of our cerebral cortex. That makes the hippocampus. The dark blue, this so-called DP, that makes our neocortex, which is the biggest part of our cerebral cortex. Then the LP and VP, those are also cortical regions, but much smaller and less well known. Then the basal ganglia is in green, yellow, and light blue, and it has these funny names, the LGE, MGE, AEP, and POA. Let's not just go into what those mean, but basically those are subdivisions of the basal ganglia. So we're looking at the stem cells and secondary progenitor cells of the telencephalon, the cortex, and basal ganglia. And then to the right are shown schematically molecular switches that control the identity of each of these regions. These molecular switches are transcription factors, which are proteins that are produced in the cytoplasm, but then are transported to the nucleus of the stem cells and neurons, and these molecular switches control the identity of the cells. So we can begin to derive a molecular logic that controls the identity of each of these regions by understanding which of the transcription factors function in these different regions. So I'll give you an example. NKX 2.1, which is this transcription factor, is expressed in the yellow and light blue regions. If you make a mutation in NKX 2.1, the yellow region no longer produces its proper output of cell types. and instead the yellow region becomes a green region. So NKX 2.1 controls the fate of an entire region of the embryonic brain, and in its absence, that region changes fate, and so that you get an extra green region and you lack the yellow region. And this type of molecular logic controlled by transcription factors is found throughout our embryonic brain and is extremely important for understanding how mutations could affect regional identity and the identity of the cells that are produced in those regions. So for instance, in our developing telencephalon, our cortex shown here in green, these would be the stem cells of the cortex, and the basal ganglia shown here in blue and red. It turns out that there are transcription factors that control the entire cortex and that are essential for making the cortex have a excitatory neuron fate. That's shown here by GUL, or glutamate. So the stem cells of the cortex are fated to give rise to excitatory neurons, and this is controlled by the transcription factors produced by those stem cells. Whereas the stem cells of the basal ganglia are fated to become either GABAergic, that's the inhibitory neurotransmitter, or a cholinergic, producing the neurotransmitter acetylcholine by the combinatorial functions of transcription factors that control cell fate in the progenitor cells and maintain cell fate in the neurons. Now we're going to address migration of cells as they leave the place where they're produced and migrate to the destination where they're needed. There are two major kinds of migration. The first is called radial migration, and that's shown schematically in this picture. In panel B, we can see in the cortex the green cortical stem cells. Once they produce the excitatory neurons, those neurons migrate radially out from the progenitor zone and into the overlying cortical plate. That's shown by these green lines that are drawn to show a radial migration pathway. The same thing happens in the basal ganglia for migration to produce the caudate and putamen, which is our striatum, and globus pallidus, and these basomagnocellular nuclei that have a lot of the cholinergic neurons that are shown here in red. So radial migration creates a map of the neurons that is very much related to the map of the progenitor cells that they were produced because of a almost straight line radial migration. In the human, compared to the mouse, the system is very similar, but there are some important differences. The mouse is shown in panel A, and the human is shown in panel B. The stem cells are red. They're down in the ventricular zone. And then the secondary progenitors are here in the subventricular zone. And then the stem cells are specially designed to produce long processes called radial glial processes that extend all the way from the ventricular zone to the outer surface of the brain where our meninges are. And radial migration involves the movement of the newly born neurons along these radial guide fibers, taking them to their proper destination so that they are correctly directed to go to their the correct location in the cortex. In the human, one of the major differences is that the size is much bigger than in a rodent brain, and these radial glia are very long and sometimes curved, as shown in this picture. And these neurons have to migrate very long distances. It can take weeks for them to migrate. It's a long process. It's very sensitive to the environment that the brain is in. Another big difference between the human and lower mammals is that the subventricular zone has many different kinds of progenitor cells, and it even has some stem-like cells that are called outer radial glia. And these outer radial glia, like the inner radial glia, send their radial processes all the way to the meninges. Now, the other kind of migration that's distinct from radial migration is called tangential migration, and that's shown schematically in this picture. And it's particularly prominent in cells that are produced in the basal ganglia of the embryonic brain. So, for instance, cells that are produced in this region, the basal ganglia, they will migrate, take a turn here, and enter the cortex and migrate as far as the hippocampus, as well as have it coming from this region. This unorthodox type of migration turns out to be used for the other major cell type, neuronal cell type of the cerebral cortex, the inhibitory neurons. So here's a picture showing stem cells of the cortex making excitatory neurons, and the stem cells of the basal ganglia making inhibitory neurons. In panel B, radial migration of these stem cells will produce the nuclei of the basal ganglia, as well as the excitatory neurons of the cerebral cortex. On the other hand, tangential migration, largely from the basal ganglia, will produce inhibitory neurons that migrate into the cortex and hippocampus, shown here in C. So we've now seen how the production of neurons in different places of the brain lead to different types of regions and cell types. And in some cases, cell types, for instance, in the cortex, are produced by different subdivisions. The excitatory neurons are produced by the cortical stem cells, whereas the inhibitory neurons are produced by the basal ganglia stem cells. And as these neurons migrate into the proper region, they begin to form their circuits, both the local circuits and the long-range circuits. So, for instance, in the cerebral cortex, we come back to the slide we saw at the beginning, the pyramidal cell, which is the excitatory neuron, is innervated. That means synapses are formed onto it from all these different kinds of inhibitory neurons that, remember, are produced in the basal ganglia. And so again, here's our blue basket cell with its axon synapsing on the cell body of the pyramidal cell, whereas our neurogliaform cell is sending an axon onto the dendrite of the excitatory cell. And this very interesting cell, the chandelier cell down here in blue, sends its axon and its synapses onto the axon of the excitatory cell. So these intricate and precise local circuits of the brain are essential for controlling how activity is regulated within small regions of our cerebral cortex. Long-distance pathways are formed by migrations of the axons that are growing out of projection neurons. And in this picture, I just wanted to show you 11 examples of long-distance pathways that are generated during development of the brain. And you can notice how extended the growth of the axons must be as they begin, for instance, for pathway 1 in this position, and grow all the way back to the posterior end of the cerebral cortex. All of these 11 pathways are what we call transverse pathways because they're growing perpendicular to the long axis of the brain. In contrast, there are longitudinal pathways which grow along the long axis of the brain. This is an interesting one showing here that begins in the eye, and the axons from the retinal ganglion cells in our retina will grow from the retina through the hypothalamus, losing my arrow. Let's see. There's my arrow. Okay, so the axon is growing from the retinal ganglion cell through the hypothalamus into the diencephalon and midbrain. So this is a very long axon tract, and at various off-ramps, a branch from the axon will grow and innervate different nuclei. And what's so interesting about this pathway is that each of these side branches innervates nuclei relating to vision and controlling vision. And this is very characteristic of our long tracts, is that not only do they end up going to final destination shown here in the superior colliculus, but they're also sending branches to way stations, each of which regulates different aspects of visual perception. Another important long tract that is highly relevant for mental disorders is the corpus callosum. Corpus callosum is the long tract that connects the left side to the right side of our cerebral cortex. And there's evidence in autism that disorders that lead to a failure to form the corpus callosum promotes autistic traits. Therefore, understanding the basic mechanisms of how axons grow long distances and then find their targets is a central issue for thinking about molecular and cellular mechanisms of mental disorders. Okay, so now we'll address synapse formation and function. This is a schematic high magnification view of a synapse. Most neurons communicate not through directly touching each other, but by sending signals across a very small gap from the sending cell to the receiving cell. And this very small gap is called the synapse. And that's shown in this picture here. So in green would be the axon from the sending cell. And at the termini of the axon, there are these little packets shown here by the circles with the orange balls inside. Those would be the neurotransmitter vesicles. And when activity comes down the axon, it then leads to the fusion of the neurotransmitter vesicle, releasing the chemical substances called neurotransmitters into the synapse, where they then diffuse and bind to receptors on the postsynaptic side, in this case, the dendrite, and a special part of the dendrite called the dendritic spine, which is where much of the excitatory neurotransmitters are released in the cerebral cortex. And so we have here neurotransmitter vesicles releasing probably the excitatory neurotransmitter glutamate, where it then binds to various channels such as the NMDA channel and AMPA channel, and then that excites the postsynaptic cell. Work in autism has suggested that many of the genes that can increase the risk of having autism are genes that encode proteins that are required for the formation of synapses and the function of the synapse. For instance, mutations in the Neurexin proteins, shown here in orange, and their friend on the other side of the synapse called neuroligin, mutations of this class of cell adhesion molecules is highly implicated in autism. Likewise, a molecule called SHANK3, which interacts with the number of molecules on the postsynaptic side of the synapse, is also highly implicated in autism. Once information from the synapse is spread all the way down to the cell body and the nucleus of the cell, that information then leads to longer-term changes in the biology of that cell, and those changes are affected by the functions of the transcription factors in the nucleus. A very famous one is called MECP2, and this transcription factor, when mutated, causes a severe neurodevelopmental disorder called Rett syndrome. And there's evidence that activity at the synapse can converge on the function of MECP2 in the nucleus, thereby connecting how activity of our brain leads to long-term changes in the biology of our neurons. And mutations in MECP2, of course, cause very severe problems in mental function. There is an interesting result, though, from Adrian Bird's lab, and he showed that in a mouse that had the MECP2 mutation and was showing the developmental effects of this, that if he then restores the function of MECP2 in this mouse later in its development, that a lot of the problems that the mouse was having are reversed, leading to some optimism that many of the symptoms that we observe in some cases of neurodevelopmental disorders have the chance of being reversed if we can understand the fundamental molecular and cellular defects that are causing them. Okay, now we're at the bottom of our neurodevelopmental steps, and this is where the other kinds of cells in our brain play critical functions in establishing brain function and physiology. And I don't have any pictures of them. I just want to point out that these cells include the cells that form myelin, which is a insulating sheath along axons that increases the speed of neurotransmission. It includes the astrocytes, which is among the most numerous cells in our brain, which perform all sorts of critical functions, including maintenance of the function of our synapses. It includes blood vessels in our brain, which of course are essential for bringing in nutrients and removing toxins from our brain, and the meninges of our brain, which perform all sorts of protective and nutrient functions as well. Okay, now I'm going to switch a little bit and just use some of the information that I've been describing to propose some neurodevelopmental models that could explain some forms of childhood and early life mental disorders. So the first one I call a detection of salient synaptic information, or signal noise processing. And it depends upon establishing the correct balance of excitation to inhibition in our local circuits as well as in the distributed circuits. Our brain is much like a signal detection device, and if that device is noisy, it's very difficult for the brain to distinguish salient important information from local background noise. Epilepsy is a classic example of a neurophysiological state in which there is excess noise in our brain, preventing optimal brain function. And it's known that in autism, about 30 to 40% of patients at some point in their life develop epilepsy, suggesting that in those patients that have this problem, signal-to-noise processing may be abnormal and may be contributing to some component or components of their problems. This is a picture of a bell curve schematically describing optimal function curve for a local circuit. So in the middle is the maximum height of this curve, and that's where optimal function is taking place. And then going to either the left or right side is suboptimal function, and that can be created on the left by too little signal. So there's too little signal coming into the system, you're not going to have much function. For instance, if the axon tracks providing information to a region are not present or not functioning well, you're not going to have much input and therefore very little signal. On the right side, however, you have suboptimal function if there's too much noise. And that can be due to a problem of too little inhibition within the system. So those local circuit neurons that I was describing, perhaps if their function is suboptimal, you'll have too little inhibition, and therefore your balance of excitation inhibition will be too high. You'll have too much noise, and you'll have less function, and you'll be susceptible to seizures. So, for instance, changes in the numbers or functions of the GABAergic, which are the inhibitory interneurons, could affect the tuning curve. There's a good example of this in experimental mice. Mice that have a mutation in a channel called SCN1A. That's a sodium channel that's essential for the action potential in the axons. And mice that are missing only one copy of the SCN1A gene have behaviors that are reminiscent in some degree of autism. Of course, mice don't have autism. They don't have the brains of humans, and we can only use surrogate behavioral assays, which are quite crude, but nonetheless provide some insights into perhaps autistic-like assays that probe possible autistic-like behaviors. In any case, these mice have problems with social behavior and anxiety, and Dr. Catterall's lab has worked a great deal on this to point out that humans that have mutation of this gene have epilepsy, cognitive dysfunction, and autistic behaviors. It's a syndrome called Dravet syndrome. And Dr. Catterall's lab found that he could improve the function of the mice by giving them a medicine called clonazepam, which functions to activate the inhibitory system in the way that the neurotransmitter GABA does. So he was able to show that treatment of mice that have too much excitation, too little inhibition, with a GABA-like compound can rescue several of the behaviors of the mutant mouse, providing some hope that if we understand which patients have a defect in this type of molecular system, that we would now have a reasonable idea for a medication that could help them. I should point out that there are also examples in schizophrenia of defects in the balance of excitation and inhibition. Work from Ted Jones' lab originally, and work from David Lewis and the Venice lab and several other labs have shown that in almost all patients who have died who have schizophrenia, that the level of the enzyme that produces the inhibitory neurotransmitter GABA, this enzyme is called GAD, the level of GAD is reduced in, I believe, all of the reported studies. There's also evidence that there are defects in the excitatory neurons as well, that their size, the cell body is reduced, and that their dendrites, which are receiving excitatory input, don't have many of these specialized receiving structures called spines. So there's a great deal of interest in schizophrenia now to understand the nature of these neuropathological findings and how that relates to the functions of their local and extended circuits. Okay, the second model that I just want to briefly mention is one in which there's too much growth of the cerebral cortex during development due to increased growth factor signaling. I bring this one up because it's seen in autism that a significant fraction of the children early in life have a cortex that's about, I think, 10% larger than normal. And this is observed for several years of age, and then I think around 7 to 10 years of age, the size of the cortex becomes more similar to children who don't have autism. So it's been a mystery why there is this enlargement of the cerebral cortex in so many of the children, and it may provide a useful clue about some of the underlying cellular and molecular mechanisms that are abnormal in their brains. I should point out that some of these growth factors that I talked about earlier, particularly the ones here in blue, the FGFs that are produced by the rostral patterning center, these can control the size of the brain, as well as the other growth factors can as well on this picture. And so I know our lab and other laboratories are quite interested in exploring how growth factor signaling can control brain size and explore its relationship to autism. One of the key regulators of growth factor signaling is a gene called P10, which is mutated in many of the children who have P10 mutations have autism. And so this is an interesting inroad into understanding how growth factor signaling can control brain size and therefore autism. The third model I want to point out is a model in which there's dysfunction of long-range circuits, as exemplified by the cortical basal ganglia circuit shown in this picture, where the cerebral cortex excitatory neurons shown here by green axons leave the cerebral cortex and enter the striatum, where they synapse on the GABAergic projection neurons of the striatum, who then subsequently send their GABAergic projections into the brainstem. Then there is a reciprocal back projection from the brainstem to the cortex, as shown here by the blue and pink axons. This is a nice example of an extended complex long-range circuit that's part of a critical, call a brain machine involved with cognition. Mice that have mutations in SHANK3, which is one of the proteins I showed you in the synapse picture. This is that protein in the postsynaptic density that interacts with the ligands, also interacts, I believe, with the glutamate receptors. These mice display autistic-like behaviors, striatal dysfunction, work from Dr. Feng's lab. And SHANK3, again, is a gene, when mutated, increases the risk for autism. SHANK3 is expressed very highly by the basal ganglia in the nucleus called the striatum, and it regulates the assembly of the excitatory receptors, the glutamate receptors. And they found that the receptor subunits shown here, glutamate receptor R2 and NR2A and NR2B, are reduced in striatal synapses in SHANK3 mutant mice, showing that excitatory inputs into the striatum will be inefficient if you're lacking the SHANK3 gene. I think that in schizophrenia, it's very likely that there are defects in this long-range circuit that we're talking about, particularly from the prefrontal cortex to the basal ganglia and to the thalamus. And in imaging studies in schizophrenia have shown that the prefrontal cortex can be small, hypoplastic, and has hypofunction or reduced function or activity. There's probably abnormalities in interneurons in the cerebral cortex, as evidenced, as I mentioned, by the reduced expression of the enzyme that makes GABA, called GAD, G-A-D, and the projection neurons in the cerebral cortex have defects such as reduced GABA receptor expression and hypoplastic spines on their dendrites. In addition, there's imaging studies that show that the striatum is small or hypoplastic, as well as the mediodorsal nucleus of the thalamus, which is the critical nucleus that sends projections to our prefrontal cortex. And the final model I will just briefly mention is that it's in child psychiatry, it's well known that many of the disorders that are seen are highly enriched in boys. This is particularly true in autism, in ADD, attention deficit disorder. And so there's something about the male brain or the male genotype, which is basically the Y chromosome, which I believe predisposes the developing brain to these disorders. So it could be genes expressed by the Y chromosome, or it could be the sex steroids that are different between boys and girls. So perhaps the female brain is protected from the genetic and environmental influences that lead to this 4-to-1 ratio of boys to girls in autism and other childhood disorders. I should point out that this idea is not unique. There are several other excellent scientists who have proposed this idea prior to me bringing this up. So in summary, we've reviewed sequentially the major steps that are required in making the brain. And they're listed here again. I won't repeat it. And I also want to have you come away with the idea that we can't just understand the neural defects by thinking about one neuron at a time. We have to think about neural circuits and how disruptions of a given neuron within that circuit can lead to a distributed effect that derails normal functions. That's it for my presentation. I'm happy to answer any questions.
I think Dr. Bornstein's microphone is muted.
Okay.
Oh, hello? Oh,
Yes,
hi.
yes.
Oh, hi, John. I just want to thank you for really an outstanding presentation with a lot of information that shows how complicated the development of our brain is, but shows the promise of what research can do moving forward. We have time for one brief question that a few people have asked in different ways, and that is, are there any steps that a person who is getting ready to become pregnant or is pregnant, are there any protective steps that they can take to increase the likelihood that this complicated process goes well?
Yes, I think there's some very easy and important things to do while you're pregnant. Good nutrition, of course, is absolutely important. And good nutrition includes avoiding substances that can be toxic to the brain, both developing and Forebrain, substances such as alcohol and drugs, various, of course, non-prescription drugs, street drugs, as well as we have to be careful about certain prescription drugs, so you need to discuss with your physicians about what you're taking during your pregnancy.
Okay, thank you. Our time is just about up, so I want to say, Dr. Rubenstein, thank you so much for your presentation, and more importantly, thank you for the work that you've been doing to help us better understand the brain and develop new treatments for brain illnesses. I also want to thank our audience for joining us today. The Foundation, through its research grants, is dedicated to improving the lives of people with a wide variety of psychiatric conditions. Thank you for joining us on that mission. All of the research we fund is made possible through private donations. So if you'd like to make a gift, please visit our website, bbrfoundation.org, or call 1-800-829-8289. This webinar has been recorded. If you've missed any portion of it or would like to share it with a family or friend, please visit the webinar page at our website. I hope you'll join us again next month as we hear from a different scientist, Dr. Cameron Carter, professor in the Department of Psychiatry and Psychology at the University of California, Davis. He will speak to us about brain networks in schizophrenia and bipolar disorder, targets for novel therapies. That will take place on Tuesday, June 10th at 2 p.m. Eastern Time. Thank you all, and enjoy the rest of the day. Take care.